All right, so here we are with chapter number 2. Chapter number 2 is all about cells. Now, living cells are the fundamental units and the simplest units of life. They are hundreds of millions of biological molecules that kind of come together to make a cell. There are two major types of cells, prokaryotic cells and eukaryotic cells, and that's something that terminology that you're probably familiar with from early cell biology class. Now, understanding of the cellular structure is going to be of fundamental importance and will help us understand the biochemical reactions that take place within living cells. And that's going to be like, what are the reactions that are capable of taking place within one type of cell versus a different type of cell? And think about that broadly, as in, like, are there certain reactions that can take place in eukaryotic cells that cannot take place in prokaryotic cells? And the answer is yes, because you have that membrane-bound organelle breakdown. Now, there are a total of 5 major important themes regarding cells. First, water. We're going to be discussing the role of water and what water does. Cells have biological membranes. They engage in self-assembly. They are molecular machines, and they are involved in signal transduction cascades. First and foremost, water. H2O. Water consists of two hydrogens and 1 oxygen. Now, hydrophilic molecules will dissolve in water because they're going to be able to establish bonding with the atoms of water. Hydrophobic molecules, on the other hand, are not going to dissolve, but they're going to actually aggregate together. We see that illustrated below. In model A, we see our hydrophilic molecules, which are able to freely interact with water and remain separate from one another. As you can see, I believe I've got my, no, I don't. Maybe I do. Just a second. Okay, there we are. This subject, this one, and this one. Now, in model B, we've got hydrophobic molecules. These 3 large red molecules are aggregating together because of the fact that they're not going to be able to freely interact with water. Now, amphipathic molecules, for example, phospholipids, are going to form bilayers. Amphipathic molecules have a region that can interact with water and then a region that cannot. Protein and DNA folding are stabilized by these sorts of interactions. So they're going to be stabilized by hydrophobic and hydrophilic interactions. Now, biological membranes are thin and very flexible structures. These are as an example, lipid bilayer. Here what we have is a head group that interacts with water, so that would be hydrophilic. And then this inner region here, and let me go ahead and point out the other hydrophilic region, where I've drawn this little half of a box, that's your hydrophobic region. These are portions of these molecules that interact with one another and are stabilized by those interactions. Overall, this structure is going to provide a physical barrier between two different portions of a cell, or basically separate material inside a cell from material outside of a cell. Self-assembly is when biological molecules undergo kind of a spontaneous organization into a supramolecular structure. So an example of that, a great example of that, is your ribosome. Ribosome is where RNA is translated into protein. And so here what we have illustrated is the small and the large subunits of a ribosome, and then what we've got with this red line is our messenger RNA, and then this blue line right here is our resulting protein that is translated from at the ribosome. Now, molecular machines are many subunit complexes that have moving parts and they perform some sort of work. So whether that's translation, it could also be migration along an actin filament within a cell. These are going to undergo some sort of, or they're going to carry out some sort of work to move, one object to another part of a cell, or one object from one part of a cell to another part of a cell. Molecular machines are going to convert energy into directed motion using the energy changes in the 3D shapes of proteins. So an example of that, hydrolysis of ATP or GTP. So these two molecules, ATP and GTP, adenosine triphosphate and guanosine triphosphate, can be hydrolyzed into ADP, adenosine diphosphate, and GDP, not gross domestic product, but instead guanosine diphosphate. Signal transduction is the process that organisms use to receive and interpret information or signals. So an example would be a physical stimuli, and that could be light, it could be presence of oxygen, it could be sound, any number of different things. And what's going to happen is a protein is going to receive that and transmit a signal to another part of the cell, or transmit a signal so that, to basically entities that do not, or that cannot perceive that themselves. Organisms can only adapt to changing conditions if they can recognize, interpret, and respond to that message. So simple recognition isn't enough. Recognition and interpretation is not enough. You also have to be able to respond. So the sequence of events would be reception, transduction, and a response. So you receive the cell, you transmit that you receive the signal, you transmit that signal, and then the cell does something in response to that signal. All right, I'll see you in the next part.